Okay, I guess it's just about 10:30 now. So we're gonna get started with the third lecture in our series. So Natalia, if you want to take it away.
Thank you very much. So today I will talk about mathematical modeling of drug resistance in cancer. So some of this stuff is a little bit older and it appears in this book, but also there'll be some newer stuff, more applied. So I will first talk about the specific type of resistance that we have been working on, familiarize you with some mathematical models, how to approach this problem. And I will present an application to a specific cancer, which I would say, I think it was sort of a success story in the sense that we were able to participate in some decisions that were made in terms of treatment. So let's see. As you, of course, know, resistance is one of the most difficult problems in public health, and it occurs everywhere. Drug resistance is seen in tuberculosis, malaria, HIV. It occurs when bacteria, viruses, fungi, or parasites evolve mechanisms to survive and resist a treatment that previously worked. Happens all the time. So there's resistance to antibiotics, there's resistance to antiviral, antifungal drugs. There is resistance to pesticides in agriculture, which ruins the crops. And of course, today, I will talk about resistance to cancer drugs, because this is relevant to the topic of this conference. And so this is a big field in itself. So there are different ways in which cancers evolve resistance to drugs. So here I list some of these types, and there are more. So the first one is pharmacokinetic resistance, where drug just simply doesn't get delivered to cancer. I think one of the problems that I'm familiar with in this respect is the delivery of oncolytic viruses to the tumor, and there are other ones as well. The second one is drug efflux pumps. Cells learn to extract the drug, pump it out, so they don't get killed by the drug. Phenotypic plasticity, including EMT. So cells, without changing their genetic makeup, can adapt and do something in such a way that drugs can't get them. Another way for them to hide is to become stem-like. Sometimes stem cells are not susceptible to some of the drugs. And finally, genetic and epigenetic resistance. This is the one I'm going to talk about because I do evolution. So this last type here is a change that cells acquire, and that change is inherited when the cell divides. So that's the thing that characterizes this type of resistance. It can be genetic or epigenetic, but inheritable, right? Heritable by cells. So I will talk about targeted cancer drugs, because for those, this mechanism is especially important. So the traditional drugs, the traditional chemotherapy is toxic. It's an agent that may kill all dividing cells, horrible side effects. Targeted drugs are small molecule inhibitors. It's a new generation of drugs, only been around for about 15, 20 years. These drugs target the pathways that make cancerous cells cancerous. So they are able to determine which cells need to be killed. So the first example is the drug called imatinib that was targeted for CML. CML is a leukemia, and I'll tell you much more about it. After imatinib, they invented a whole lot of different such drugs, which are used both for CML and other cancers, but it all kind of started with leukemias. So why do we model this mathematically? So I think the simple answer to this question is that development of drug resistance is a complex evolutionary process. And you cannot just kind of figure it out by talking about it and drawing diagrams, right? We need specific models for that. And by using these models, we can address many questions that arise in this respect. So I'll list some of them here. So drug resistance and drug combination therapy, optimizing combination strategies, the problem of cross-resistance, and cyclic therapy. So these are the ones that I'll mention today. But first, let me show you how we do this. So let's suppose that we have a cancer cell. So what can it do? In this model, we assume that it can divide with a rate L, and it can die with a rate D. So this is what they call a branching process in mathematics. So you can see that a kind of a tree of divisions grows and also gets depleted by the process of cell death. So in the absence of treatment, we assume that the rate of division is greater than the rate of death, and then the colony grows exponentially. So exponential growth is probably a good assumption for leukemias. It's not a good assumption for solid tumors. So most of what is of the stuff will be applicable for leukemias. So imatinib, let's suppose we apply drug. So now the death rate of cells increases, and we'll call it H, the additional death rate that is induced by the drug. So now, if this new death rate is greater than the division rate, the colony will shrink and eventually disappear. And that would be a successful treatment. Okay, so in best case scenario, this is what happens, and this is ideal. Unfortunately, it's not always like this. So just to show you a few slides on stochastic modeling, how you approach it. This is pretty standard. So we assume a linear birth-death process. So in this simplest diagram, I only had divisions and death and only one type of cell. We can calculate the probability to have I cells at time t. We can write down a balance of probabilities, which translates into Kolmogorov forward equation. This contains all the information. So useful information that we can extract consists of, for example, expectations and moments for the number of cells at the given moments of time. Or we can use the formalism of probability generating function, which is very convenient here. This infinite number of nonlinear ODEs corresponds to one PDE, first order PDE, which you can solve. And this, for example, gives you directly the probability of extinction. So probability of extinction here is the same as treatment success, right? So that's what we're after. So this process is kind of at the core of many calculations here. But of course, there are many bells and whistles that go on top of this to add some realistic phenomena. So my case study is leukemia. I already showed you this slide. And I'll talk about two leukemias. One is CML, and the other one is CLL. And we'll start, they have different cells of origin. So this CML here stands for chronic myelogenous leukemia. It's not a very common disease compared to some other more common leukemias, about 9,000 new cases in the US each year. It's an adult disease, it's not a childhood leukemia. So the origins of this disease are well understood. And I think many of you probably know this, but I'll still mention it because it doesn't stop surprising me how weird this is. So look at this diagram. So it shows two chromosomes. This is chromosome 9, and this is chromosome 22. You know that human chromosomes are numbered in the order of their length, right? So it becomes shorter and shorter. So look at these chromosomes. And so this one breaks here, and this one breaks here, exactly at these locations. These two pieces come together and fuse at exactly a specified location to create what is called the Philadelphia chromosome. If they break somewhere else and fuse there, nothing happens. So they have to break at an exact location and fuse exactly there. And then they create this gene called BCR-able gene. Sorry, when I said chromosomes, so they have to break exactly inside the gene BCR and here inside the gene able, right? So it's very specific location inside these genes. So by fusing these two, we create a BCR-able gene, and that's the one that starts leukemia. How likely is this, right? How likely is it to happen? But nonetheless, it happens that many times a year, right? So this is the origin of this cancer. And this BCR-able fused gene creates a protein, and the drug imatinib specifically is designed to bind that. And this results in the death of the cancerous cell. If there is a mutation in this BCR-able protein, the drug imatinib can no longer bind, and that is treatment failure, right? So that's how it is. So a mutation in this fused gene, which makes cancerous cells cancerous, makes it resistant to the drug. So this is the problem we are going to deal with. And here's a simple diagram of how we can view the process of treatment and resistance. This is not a simulation, it's just a schematic. So cancerous colony grows, and we start treatment. And in the best case scenario, the colony stochastically shrinks and is gone. Now, as the colony divides, it undergoes cell divisions, many cell divisions. Every time a cell divides, so these are cancerous cells, BCR-able, they have the BCR-able gene. Every time a cell divides, there is a small chance that there is a mutation that makes the cells resistant to cancer. So this is shown in these little bubbles. So these bubbles appear, and maybe stochastically they then die because there is a birth-death process, you know. But the one that doesn't die, you start treatment, it's there, and treatment fails. Because when you start treatment, this colony is not susceptible, and if it's a successful colony, it just grows. So that's treatment failure because of resistance. So how do we incorporate resistance in the mathematical model? So this is the first part that I showed you. Now, we're going to assume that this kind of division happens with rate L, the rate of divisions, times 1 minus U. And U is the probability of mutation. With probability LU, this division is not faithful, and a mistake is made. This is the resistant cell. It happens at a small rate, and it has a death rate and a division rate, right? And then when we start treatment with imatinib, these cells are susceptible, and their death rate increases. These cells are resistant. Their death rate either doesn't change or doesn't change enough, right? It continues to grow. So the colony of these cells will shrink. This one will continue growing. So that's the failure of treatment. So how can we prevent this process? This is a completely natural evolutionary process that happens in the expanding colony of cells. So what can we do about it, right? The answer is nothing much. It's going to happen because these are mutations, right? So, but what people do and how they deal with this in other cases. So inspiration comes from the, HIV and how it's dealt with there. So when the first HIV drugs appeared, they were failing because of resistance. And that's a very different disease, right? It's a viral disease, and it's evolution of viruses. So those viruses mutate and they stopped responding to the drugs. So what they did, they started giving patients a cocktail of drugs. So the standard of care now is combining 3 drugs. Why does it help? Because if a virus evolves resistance to one of the drugs, it's probably still susceptible to the other 2. And it takes 2 different mutations, independent mutations, to evolve resistance to 2 drugs, and 3 independent mutations to evolve resistance to 3 drugs. So this is much less likely. It still may happen, but the probability is a lot lower. This is what we tried to do to apply this concept to treatment of cancer. And let me dwell a little bit on this. This is a combinatorial network, mutation network of cancer of evolution of resistance. Let's start here. Okay, so let's start here. This is a type that is fully susceptible to all the drugs. So we're going to assume that there are 3 drugs that we are dealing with, and this cell is fully susceptible. You apply any of the 3 drugs, it responds and it dies off. So there are 3 ways in which it can develop a dangerous mutation. With probability U1, with the rate U1, it may become resistant to the first of the drugs. So if you can see, this is, we're using binary indices. So this is 0, 0, 0. It means that it's susceptible to all the 3 drugs. This is 1, 0, 0. It's resistant to the first of the drugs, but it will still be eliminated by the 3 drugs because the other 2 drugs will do the job, right? So this cell is resistant to the second drug, but susceptible to the first and the third. And this cell is resistant. I think it's supposed to be, oh yeah, here, 0, 0, 1. So it's resistant to the third drug. So these 3 types of cells are resistant to one of the 3 drugs, but they are killed by the other 2. Then you can continue mutating. And so this cell can acquire a second mutation. Now it's resistant to drugs 1 and 2. Or it can acquire this mutation, and it's now resistant to drugs 1 and 3. And so on. So there are 3 types of cells that are resistant to 2 of the 3 drugs. And finally, this is a culprit. This one is fully resistant. So if there is a cell like this in the system, it will not die, and the treatment will fail. If it's not there, the treatment will be successful, most likely, right? So, if these mutation rates are very small, the likelihood of this happening is very low. The likelihood of this happening is much higher because there's only one mutational step that separates this type from the wild type. So it's very likely that these are already there even when you start treatment. These are difficult to evolve. This is how drug combination helps avoid resistance. And this is a very general principle, so starting with the HIV treatment, so this can be also applied to cancer. So what's the main problem with this? There are several problems. One being that we need to have the 3 drugs. If there's only one drug that's been invented, there's nothing much you can do. And the second problem is side effects. So we'll talk about both. So we've been working for several years on stochastic analysis on combinatorial mutation networks. So this is a combinatorial mutation network. So this kind of in spirit is similar to the work of Lurie and Delbruck. So maybe you know that these are Nobel Prize winners in 1969. They did something completely different. They were studying bacteria. They set off to prove that evolution doesn't have a direction. Like evolution is random. Not in Eduardo's talk, but the randomness of evolution. So they grew bacteria, and after a certain amount of time, they counted the number of mutants, and they developed a mathematical model that describes that. So since their work in the following 50, 60 years, a lot of mathematical methodologies have been developed to deal with this problem. So our problem is kind of like this, but it's adapted to cancers. So cells can die. So usually in this framework, they don't die. And we don't just have one mutation, but we have this mutation network. So multiple mutations. Right? So it's a more complicated problem. So let's see. So we want to know the distribution of mutations after a while. As a colony grows, is it resistant? Right? This is one of the questions that we want to ask. So we want to count mutations. Or we want to determine the probability of not having a resistant mutation. So how does resistant generation depend on cell turnover? Cell turnover is a ratio between division rate and death rate. So a low turnover colony is a colony that grows and cells rarely die. High turnover colony is when cells die all the time, right? But they still grow. So there is a difference. And so the first question, so for very low turnover, or 0 death, how many cell divisions does it take to grow to size n? Let me ask you this question. How many cell divisions does it take for one cell to grow to a size 1,000? How many cell divisions? They don't die. So 999 divisions, right? Because every time a cell divides, a new cell is created. So 999 cell divisions separate one cell from a colony of 1,000 cells. For a high turnover colony, when cells die all the time, how many cell divisions does it take? Many, many more, right? Because they die all the time. So they have to, it goes back and forth. So for a low turnover colony, there are fewer divisions that create 1,000 cells compared to high turnover. So when we're talking about resistance generation, and resistance cells are generated by divisions, where are we more likely to see a resistant cell? In this type of colony or in that type of colony? So a naive answer to this question is, of course, here, because there are more cell divisions. So in a colony of a given size, they have undergone more divisions, there's going to be more resistance. But it's not so, right? So if you do the math of the way I described, you can prove that the answer to this question does not depend on turnover. In both a colony that doesn't die, and a colony that divides and dies, there's going to be the same probability of developing resistance. And this is because this is shown here graphically. So there are many more cell divisions here because they're balanced by cell death. So the resistant colonies are created many more times during this process, but the death rate is higher. So they also die more often. And the two, it turns out, balance each other completely in this case. So more resistant colonies are created, but they die more often. The result is the same. So a measure of resistance generation can be defined as N of failure. What's the population size when the probability of resistance is given by a threshold? For example, I don't know, 10%. So the N of failure is constant for one drug treatment for any turnover. These are the cells that never die. These are the cells that die all the time. The resistance behaves in the same way. But if we add more drugs, now the calculation is a little harder and the result is less intuitive. But the answer is that now it depends on whether you have a high turnover colony or a low turnover colony. So for multiple drugs, treating low turnover cancers is easier. So you add more drugs and N of failure increases. So this is 10 to the 8, 10 to the 10, 10 to the 12. So if you treat with 5 drugs, these are arbitrary parameters. So if you add, as you add more drugs, it takes a bigger colony size to generate resistance, right? But for low turnover cancers, actually not so good. You keep adding drugs, but it doesn't really make so much difference anymore, right? So there's a subtle difference there. So for high turnover drugs, combination therapy contributes little to preventing resistance, and it's very difficult to treat. So that's a theoretical prediction. So there are many things that you can do with this formalism. Like you can calculate how many drugs, given the mutation rates, right? We need the parameters. Given the mutation rates, how many drugs do you need to achieve, say, a success rate of 99% for a tumor of a given size? So the success rate is never 100% because there's always a chance that you create a mutant that's resistant to everything. So you can see, like here, you increase the mutation rate and you increase tumor size. You only need like 2 drugs, and if the mutation rate is high and the tumor size is low, it's not so great. So you can reduce the number of drugs using this diagram. So this paper says that basically, let's start here. Let's suppose that the tumor size is 10 to the 13, and we have a certain mutation rate. We start treatment, the tumor size goes down, and then as you reach this boundary, you can reduce the number of drugs from 4 to 3, and then from 3 to 2, and the probability of resistance doesn't decrease. So there are strategies that you can calculate mathematically that kind of keep the probability of resistance constant, but allow you to reduce the number of drugs that you apply. Okay, and so in this case, we showed that the strategy that you find, it strongly depends on the mutation rate, but it's something that you can calculate or find out somehow. And it doesn't depend on the tumor size or turnover rate, so which is great. Cross resistance. So that's a huge, question?
Just to understand the non-intuitive part where the turnover is high, more drugs don't affect that much.
Right.
So could you give it, I mean, the way I was thinking kind of doesn't make sense in my head in the sense you have, in the other, the only way you can get 2 mutations, I'm not thinking 3 right now.
Just 2.
Is that the first clone has to get big enough that you get the second one.
Yes.
Right? That's the way of getting 2 mutations. The only way the high turnover and low turnover could give you different answers is somehow the clone sizes are not getting that big for the first mutation in the high turnover case, right? But that means the total population size is.
No, the total population size here is controlled, right? So we are comparing the probability of developing a mutant, a doubly resistant mutant at a given population size.
Right. And somehow the clones of the single mutants are smaller there, right?
So there are 2 things that are going on: how quickly they are produced, and how likely they are to die. So for one drug, they cancel each other. For more drugs, these things don't cancel each other. And I can't remember if I even had a more intuitive explanation. Maybe we can talk about it later. Yeah, maybe. Good. So cross resistance is a huge problem. So how does this resistance work? So let's suppose that we have a cancer protein, and there are 2 drugs, drug A and drug B. They bind. And by binding to the cancer protein, they kill a cell. You can develop a mutation which changes the structure of the protein. So now, with this mutation, drug A cannot bind, but drug B can, right? So this cell is susceptible to drug B, but resistant to cell A. This mutation makes the cell resistant to drug B, but susceptible to drug A. This is the bad one. This is a mutation that prevents both of them from binding. And that's what they call cross resistance. Right? So there's a mutation that kind of makes a shortcut. So if we have this diagram, that's a shortcut. It's a mutational event that takes you from a susceptible cell to a cell that's resistant to 2 drugs simultaneously. And you can have all sorts of combinations. So for example, with 3 drugs, you can have 3 drugs that don't have any mutants that are resistant to all 3, or 2 of them can develop cross resistance. This one is the worst. This is a triply cross resistant situation where there is a single mutation. So let me just, sorry. I wanted to show you how it changes with my mutation framework. So this is drugs I and D develop cross-resistant mutations. So this is a pair of these. There are 3, and this is the bad one, right? So here in a single mutational jump, single event, you develop resistance to all 3 drugs. And the thing is that in CML leukemia, this is something that actually happens. So the first 3 drugs that were invented, the first one was imatinib, and then was dasatinib and nilotinib. Right? So that was great. And they showed some degree of pairwise cross resistance. So there were some mutants that would make them resistant to a couple of these. But there is a specific mutation, T315I, that does exactly this. It creates cells that are resistant to all 3. And for a while, that was the biggest problem in the treatment of CLL. I'm lucky to have met a person who participated in the development of the new drug called ponatinib. And that was specifically created to fight such mutations. So for years, this was the biggest unsolved problem. And so now they developed this drug. So these days, the first line therapy is always imatinib. This is kind of like give it to everybody. If it doesn't work, they go with dasatinib and nilotinib, and there's also bosutinib. This is a newer one. But patients that have this mutation, they receive this ponatinib drug. It may have slightly worse side effects, but it works. This type of calculations shows the probability of treatment success as a function of colony size for different combinations of drugs. So let's suppose that we have a colony and grows and stop at size n, and we'll say how likely are you to be able to treat it if you treat, for example, with one drug. So we assume a specific mutation rate, and with one drug, things are not so good. As the colony size grows, the probability to treat it successfully goes down. So when you have 10 to the 8, 10 to the 9, the mutation is there. It's not gonna work. If you have two non-cross-resistant drugs, then it's much better. Realistic size goes to 10 to the 13, so you're good. And 3 drugs is even better, right? But if you have cross-resistance, that makes it significantly worse. 2 drugs in the presence of a possibility of cross-resistant mutations kind of is in between. Yeah. So here there are specific kind of notations. This is imatinib, and dasatinib, nilotinib. So there are different types of combinations can result in different probability curves that you can calculate by using this method. So the last kind of theoretical approach before I start applications is cyclic drug treatments. We heard the talk about it today. Combination treatments are always, like, you can prove that combination treatments are the best, right? Mathematically, they kill as much as possible. They prevent resistance from happening and delay the development of resistance. But nobody uses them, right? Because doctors are careful. They don't want to combine drugs. They always start with one and then maybe add another one or replace it to avoid side effects. And just, it's not a common practice. So if we were to use cyclic therapies, what is the best strategy, right? So we want to find the optimal cycle duration and which drug should go first. So the treatment schedule is like the drug 1, drug 2, and so on. If you treat with drug 1, this is your combinatorial diagram. This type is resistant to that drug. The additional death rate is 0. This drug doesn't work for this one. And the second one also has its resistant cell. So this problem was solved for the first time by Day in 1986. And it became a very famous paper. So the result was like called the worst drug rule. Treat with the worst drug first. Worst means that it doesn't kill as effectively. And use the longer cycles for the worst drug. So that was the recommendation that was provided. So we tackled this problem as well. So in this old paper, they only like compared these hand-picked strategies and compared them, right? So we could do it more comprehensively. First, we discovered that there's a rule. If you have any hope of a treatment working like this, the drugs should be mutually strong. So this is an additional death rate that is given by the first and the second drug. And this sum has to be less than 1 of the inverse net growth rate of cancer. Very simple rule. If this is not satisfied, your probability of treatment goes to 0, more or less. So we designed an optimization problem. This is your the function that we want to minimize. This is basically the expected number of singly resistant cells. Okay, we don't want the double resistant cell to appear. How to minimize it? Minimize the pool of cells that produces that. And depending on delta T1, the intervals of the first drug and the ratio of the two, you can compare the drugs that start with the best drug and the worst drug, right? So you can find the maximum and the optimal strategy. The answer that we obtained was slightly different. It starts with a better drug, but uses the worst drug for longer duration. So by this more comprehensive treatment, we got a different answer. Okay, that's not too surprising. High toxicity. So if you have everything mapped out like this, right? So this diagram, it's kind of like a heat plot. So you want to minimize the probability of resistance. So you look for the hole in that landscape. And this may be difficult if there are severe side effects. So these lines are supposed to kind of represent the areas of the parameter space where you don't treat for too long and you don't treat too often, right? Because you want to kind of give the patient a break. So you don't want to be anywhere in this area of the space. So here we assume that drug A is stronger and more toxic, B is weaker and less toxic. We minimize the cycle lengths of drug A, so we're below this line. And we minimize the percentage of time drug A is used, so we're kind of at this point. So it modifies the optimal treatment. So we found the most mathematically optimal treatment and modify it by using these considerations of side effects. Okay, so we can do that, but let me just continue the story of Day and his publication. So the worst drug rule was out there, a couple of publications, and there were several clinical trials that were specifically designed to test the worst drug rule. So back in the day, they still believed mathematical biologists, right? So the multiple clinical trials to check whether this works, and all of them failed. So that was very kind of disappointing, a very disappointing result. So what we were able to understand is why they failed. Right? So there is this big parameter space of different treatment durations, the duration of drug A and drug B. And you can start with one and start with the other. The different solid and dashed lines correspond to starting with one drug and the other. Right? So at each point of this parameter space, we can compare AB, 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 or BA, BA, BA. So in this parameter, at this point, AB and AB is better. And here, BA, BA is better. Right? So what the clinical trials showed is that they picked some point in this parameter space and they obtained the result that was the opposite of the optimal result that was proposed. But this shows that it depends on exactly where you are in the parameter space. So it was a disappointing result, but it's not surprising, right? So in order to check such a theory, you really need to know all the parameters, right? And know exactly where you are in the parameter space. Otherwise, it's impossible. So this disappointing story is a lesson, actually. So the results of clinical trials have to be interpreted very carefully in terms of mathematical models. Okay, so now I'll talk about something that is more kind of real world based, and I'll talk about CLL, this a different type of leukemia, chronic lymphocytic leukemia. It's a more common cancer, and it usually develops in older patients. Most patients are diagnosed routinely during blood tests, and upon diagnosis, they do wait and see. And then when some clinical conditions are met, like something triples within a certain duration of time, treatment starts. So traditionally, patients were treated with a combination of chemotherapy and antibody therapy until the drug ibrutinib was invented in 2014. That's another small molecule inhibitor, this specific targeted drug. So in clinical trials, some strange dynamics were observed under ibrutinib treatment. So before I show you what strange dynamics are, I'll show you the normal dynamics. So this is back to CML. This is what usually happens. You start treatment, and like here, if you start treatment and the cancer decreases, if all goes well, it's like this, or like this, right? In other cases, all goes well until this happens. And of course, we already know that this is resistance, right? So this is what is normally observed. And now look at CLL with ibrutinib. You start treatment and it goes up. And then it comes down and kind of stabilizes at a non-zero level. So that is what I meant by saying something strange was observed. So patients panicked, doctors panicked, nobody knew what it was, right? So we're trying to, we were involved in this, trying to explain what's happening. And the model, so I showed you stochastic models. This is not used here because this, you'll see, you need to kind of use the smallest model to not overfit the data. So we have two compartments. One is tissues, such as lymph nodes, spleen, bone marrow, and blood. So it's a two-compartment model. So stem cells are here, and this is where blood tests are done. So we have production of cells here, migration to blood, homing when they go back. So in this compartment, we have division and growth, and most cells are here because they have the right microenvironment for divisions. In the blood, nothing divides because the microenvironment is wrong. So it's a small fraction of tumors. So after ibrutinib is applied, somehow cells appeared in blood. There was this huge blip, right, as you saw. So how can we explain this? So there are two possible scenarios that could be here. Maybe this drug causes cells to just migrate to blood, and they die there by neglect because they don't have the microenvironment, but the drug doesn't really do anything. Or maybe the drug kills them inside the bone marrow somehow. So which one? So that's what we're trying to do. So here's the model. And this is X compartments. Y is the number of cells in the blood. This is migration from one to the other. This is death in the bone marrow, death inside the blood. And this is a non-zero level to which lymphocytosis stabilizes as time goes to infinity. So that's a constant. Homing. So it's in here, right? Because, for example, if M is negative, it would be more homing than the other one. If M is negative, that means that it goes that way. If M is positive, it goes this way, right? So both processes are in M. It's a net migration. Yeah. So this is the data. We had great data. This is just part of it. See how many points we have, right? So a system of two equations with three parameters, this amount of data. So how can it go wrong? This is what we're fitting. And what we found is that there is a parameter node identifiability, which is not so bad because we were able to calculate some ratios of parameters. But there is also some weird symmetry of solutions. So there were two solutions of this linear system of ODEs, which can be solved analytically, obviously, in fit. Two solutions. One of them has a large migration and small death, and the other one is small migration and large death. And you can explain it completely by writing down the solution and doing a parameter transformations. Both fit. It's a symmetry, a real symmetry. If this was a nonlinear system, we would not be able to figure it out because we wouldn't have an analytical solution. Here we do, so we understand it. But it's really sad because we were asked to say, do they really only migrate or do they die because of the drug? And the reason for this is that there are two equations, but the data are only in the blood. So we only fit Y. So we need the data in the compartments. So we asked for this data, and this is the data they gave us. There are 2 points. Because it's expensive to go and take the cells from, but this was enough to resolve the symmetry. So we were able to distinguish between the two models. And so we were able to, so once we know which one, we have all the parameters for each of the patients. And so the result was such that the drug actually increased the death rate 5-fold in the tissue compared to the death rate without the drug. So it's a very positive result. The drug was actually working, and the migration rate was only a small part of this redistribution. So the redistribution came because this equation that sort of has this blip, but the drug was working and it was killing cells. So we were able to distinguish that. So that was a very positive result. And also we measured parameters in individual patients, which is a step towards personalized treatments. So now we can continue with this. I wanted to show you what we can actually do with individual parameters and how we can reason about resistance. So once the drug was approved, there were reports of resistance. Here and there it comes up, right? So mathematically, we can answer some questions about resistance. So 3 questions. Are resistant mutants more likely to evolve before or during therapy? Can we predict time of resistance-induced disease relapse? Well, a patient comes in, how long do I have, right? Can we predict that based on individual parameters? And if relapse is there, what can we do, right? So the first question is very simple. The question is, when do resistant mutants evolve? Do they evolve before we start treatment or during treatment? Mathematically, it's such a simple question. But I see papers over and over asking this question, right? Because it's not necessarily intuitively understood by clinicians. A very simple calculation shows that the probability of having resistance in a colony of 10 to the 12, 10 to the 13 cells is this, 0.99999. This is the probability that the resistant cell is already there when you start treatment. And intuitively, like it's a back of the envelope calculation. This is the number of cells, this is a mutation rate. Multiply one by the other, you get a number bigger than 1. It's there, right? So the resistance is there. The question is, given they're there, what happens next, right? So what does it depend on? How long does it take? So we need some parameters that are related to the 2 important rates, division and death rates of leukemic cells in patients. And that can be measured directly by using this technique that was published in 2017. Patients drink heavy water. And then they draw blood and they are able to estimate, they can calculate the net growth rate of cells, and they also can calculate specifically the division rate. So they know both independent division and death rate of cells. So each patient, and it's cheap, relatively cheap, and it's harmless. So people can do this, so these parameters can be extracted. Given this, we can predict the growth of resistant mutants as a function of time in different patients. So this is the predicted course of mean number of mutants as a function of time. See how much variety we have in the real patients, right? For example, this is very lucky. It never really comes to detection. Detection size is 10 to the 10, and for some of them, it goes very fast. So depending on these parameters, resistance can come earlier or later. The only variable are CLL growth rates and population size at detection. So the prediction is that this is the distribution of years of treatment before resistance comes up. So before 2 years, 6% of the patients will develop it before 2 years. Half of the patients will develop it before 5 years. And only 5% of the patients will not develop resistance. So this is kind of a personalized prediction that we can obtain for individual people, like how long is the expectation that in your particular case, treatment will be successful. So if prediction is optimistic, then nothing else should be done. But and you stay with monotherapy. But if it's pessimistic, 1 year or so, one should try other approaches. So what other approaches are there? So that's the last question. We checked different possibilities. So early treatment, combining of 2 treatments, or debulking. So early treatment is difficult because detection size is greater than 10 to the 9 and the resistance is already there. So if you can start treatment early, but since resistant mutants are already there, it's not going to help in this case. Combining 2 tyrosine kinase inhibitors. So it's difficult to find 2 drugs that don't have the cross resistance. And as I already explained, they're more effective for low turnover tumors, which is not necessarily the case. Debulking is a strategy that we found the most kind of hopeful here. So we first use immunotherapy, kind of decrease the number of cells, and then start ibrutinib. And let's see what happens. So if you do that, you can create a mathematical model using the methods that I described. This is a prediction in the presence of debulking by a factor of 100. So you reduce the tumor size to 1% of what it was. So it used to be 46% of the patients developed resistance in 5 years. Now it's half of that. And it used to be 5% not developing. Now it's 14%. So it really changes the numbers. And you can, this is based on the cohort of patients that we used and their parameters. So this is the prediction that we have here. And so I think that's all I wanted to say. I want to thank my group again. And yeah, we're hiring. Thank you. There's a question there.
Thank you. Oh, questions already.